Good morning, students. Today will be the continuation. So just to recall what was done in the previous class, we had discussed about the monohybrid cross, the test cross to check or to determine the genotype of the dominant phenotype. Then there is incomplete dominance, a deviation from the cross which shows the factors, the dominant factor. When it is with the recessive factor, it is incompletely dominant over the recessive. That is your incomplete dominance, also called partial dominance. Then you have understood about, based on that incomplete dominance, the concept of dominance. The recessive character, which we say the modified allele, it will have the three cases. The first case, though it is recessive, it can produce the less efficient or normal enzyme, so the transformation of substrate can occur. Or it can produce the enzyme but does not have any function, which is the recessive, not producing the function. And the third case is not producing the enzyme at all. So this is what your three types of alleles will be present, and we are talking about the concept of dominance in relation with your incomplete dominance. Then coming to your codominance. So codominance, it was your blood group IA and IB, it's together expressed, and that is called as codominance. Together they are dominating, both are expressed. So based on the blood groups, there will be several questions. So I had given a question yesterday and I have solved and given it to you to give you an idea how to go about in the cross which I gave the question for you yesterday. Today I am going to discuss what the cross is based on and what kind of result you should give. I just gave the possibilities for you. So I have taken A blood group and B blood group. So in your A blood group, you can choose any homozygous. Homozygous, I hope you remember when I say homozygous, we are saying IA. Since the question, based on the question you will come to know homozygous or heterozygous. If they don't mention anything, we have to write the possibility, that is heterozygous condition. So we have tried two cases. So if the question is asked for 5 marks, then you have to mention two cases. Case when it is homozygous, A blood group is homozygous, or A blood group is heterozygous. So like that the two cases will come. Similarly here, two cases. So don't go to explain more, only two cases are enough, maximum two, because for 5 marks only it is asked, not for 10 marks. So see that you are giving, if it is 3 marks and they have not mentioned whether it is homozygous or heterozygous, directly start with your heterozygous condition. You have to start in the beginning. There are your two cases, that is your homozygous and heterozygous condition, but we will consider in general the heterozygous condition to get the possibilities. So in case if it is possible, if it is possible to get the progeny, what is the requirement here? We have to include the gametes here. So how to produce the gametes? Segregation of the each allele. Once you have got the alleles separate, you are using your Punnett square, and in your Punnett square or the checker box, you are able to get the 4 types of blood groups based on your calculation here through your Punnett square. So there are 4 possibilities. What can you infer? 4 possibilities. What was that possibility I had written? AB blood group, B blood group, A blood group, O blood group. All are possible. Now the question can be asked in a way, what could be the percent, or just give the possibility, or the possibility if they ask is also fine, or they can directly come for give the percentage of the progeny that can form. So when I say percentage, consider this to be 100. When I say 100 and these are all one, one individual out of that 100. So when I say 1 out of 100, you can say 25% actually. So 25%, when you add up all, you will get definitely 400. So that's what we are saying. You have to divide that 100% in your progeny. So you can say 25% chances of AB blood group. The progeny will have 25% of chances of AB blood group, 25% of A blood group, that 25% of B blood group, and 25% of O blood group. This is how we are saying. Okay. So the progeny can have the following percentage, the possibility. Okay. So this is what about the numerical value which I have to say can be included in your exam. So don't leave it just as we got the progeny as all the four blood groups, that is AB, A, B and O. So if you end up there, you might lose 1 mark. Why 1 mark you are losing? Because you are not mentioning the percentage. Here it is 25% each. Okay. So in case if you want to give the ratio, yes, you are welcome to do the ratio. It is nothing but your percentile, we are saying 25% each. In the ratio form, we are saying 1:1:1:1. But remember to write which is the blood group. So A, B, AB, O. That way you have to write. Both are possible. Any one you have to write, but numerical. Either the ratio or the percentile. Okay. So see that you are following that method. So the codominance, I hope it is clear. Codominance topic, we are talking about the AB blood group, and blood groups crossing can be asked as a separate question. So when a male of blood group A marries a woman of blood group B, like that the question can be asked. So you are supposed to go in this format. Okay. So in case if they are mentioning homozygous or pure, true, so such terms, little you have to be very careful. There it is the a hint given to you, it is homozygous. Okay, today we will be starting with the next further on. So you have understood the multiple alleles, right? Multiple alleles, there is IA, IB and I. All these three will code for one gene, which is that blood group. Okay, so here in pleiotropy, one gene has more than one effect. Okay, one gene has more than one effect or multiple effect. So it controls many phenotypes. Let us understand what that pleiotropy is. So in your P plans, we are considering, okay, capital B and small b to represent production of starch grain. Starch synthesis occurs in all three cases, but based on the synthesis, even the grain size will be formed. So the grain size is large in case of the capital or the dominant. In case of the recessive, it is small. See here, it is large in the dominant, it is small in the recessive, but it is medium when it is your hybrid or heterozygous condition. So doesn't this resemble your incomplete dominance, where the offspring does not resemble to the parents? Okay, so this is your incomplete dominance. Now that was one feature. One gene is able to produce starch grain. Now one more feature is there. It can produce the shape of the seed. So produces round seeds, capital B, small b, that is recessive, reproduces wrinkled seeds. So when it is in hybrid condition, dominant is the one which is observed here. It produces round seeds even though it is hybrid. So this is your normal. The law of dominance works here. Okay, and the recessive is suppressed. Now, though the two types of actions are taking place, the main intention of explanation of this particular starch synthesis as well as the shape of the seed in pea plants, we are trying to say one gene controls two features. One gene controls two features. That's what we are trying to say here. Your single gene has more than one effect. Okay, so it can be more than one, more than two, so multiple effect. What is it doing? It controls the phenotype. Okay, so I hope it is clear. This is the explanation part. You can go through in your own time. So here, I hope it is clear. Now coming to your inheritance of two genes. We finished already inheritance of one gene, that is your monohybrid cross involving one character. Now it is dihybrid cross. We are involving two characters. So what are the two characters Mendel has started to study? The simplest which we are studying here is the color as well as the shape of the seeds. Now when I say color, there is your yellow and green. So they have given the name Y, capital Y and small y. Then there is the shape round and wrinkled. So round, R, capital R. All remember capital will be dominant. So small r for wrinkled. So they have just crossed. This is in case if you are able to write, you can write. I've just mentioned it. The explanation of the inheritance of two genes, I've started from the next slide for you. This is the important introduction. Which plants he crossed, which characters he had crossed. Pea plants that differed in two characters, that is color and the shape of the seeds. This is the cross, dihybrid cross he had done. Okay, so when I say dihybrid cross, cross involving two characters. Example, color and the shape of the seeds. I hope it is clear. This is the example. So your inheritance of two genes. This is from the textbook. This is to make you more clear. Let me show you what I have done here. I hope you can see. Let me zoom it first. Now, what did Mendel take? Pure round yellow, pure wrinkled green. So it had the respective genotype as we had said. R for round, Y for yellow, small r for wrinkled, small y for green. Okay, so capital is for dominant, small letters for recessive. Now there is only one type of gamete formed, R and Y. Now you might wonder why R and Y together they are kept. Remember, now he is talking about in this nucleus. In the nucleus there is your two different chromosomes, okay, not homologous, different, non-homologous they are. So one, let me show it another color so it doesn't confuse you. So these two are totally different chromosomes. Maybe chromosome number 1, chromosome number 3. So it has R over here, okay, and Y over here. So we are talking about, this is in diploid state, remember. So when gamete formation occurs, what happens? This duplicates, right? It duplicates to form R and R along with Y and Y. And then during the gamete formation, each will separate. So in one gamete, one R will come, one Y will come. Still confusion? Let me know. I'll just repeat it. This is why I said learn the meiosis process clearly. So your chromosome So this is your Y. So this is your diploid condition. When replication occurs, it is Y, Y, R, R. So this is in diploid condition. That is what your parents are. Okay. So don't worry. We have followed the textbook. First comes RR and then YY. So when the gametes have to separate, there is R and Y in one, another gamete R and Y in one. Okay. So I hope it is clear. So same thing I am saying, one gamete because it produces similar type of gametes. So only once if you write is enough. Even for the monohybrid cross you can do the same thing. Okay. So this is in dihybrid cross. We are saying the single type of gamete formed. When they are fused, we get the F1 progeny round and yellow. Observe the genotype. It is hybrid. When the F1 progeny is self-pollinated, so this is how you have to say self-pollination, we get, from each parent 4 types of gametes. Please mention this step. This step you have to mention, don't skip it. So there is for these, there are 4. This plant has 4 types of gamete, as well as similarly as you can find, it is the same genotype, same gametes you'll find. So there are 4 different types of gametes. Now coming to the F2 generation, how does it form? This is your Punnett square. So earlier it was only this much, right? Your two columns, 2 rows, like that it was. Now we are talking about all 4 gametes to be included. So you have to write all the gametes here, So you have to mention male and female. It doesn't matter who is male and female now here. You have to write this but to indicate the gametes of male and the female. Okay. Observe here. When we do the cross, I've written in different colors. If you can identify, I'll just draw the triangle first. So whatever the triangle I've done, all are round and yellow. Okay. Round and yellow. Now I'm going to put a tick. This is round green, and round green. Okay. Now a circle I'll put. This is wrinkled yellow, and wrinkled yellow, and finally the wrinkled green. It's only one. So if you count, there are, see, the best way to draw this box is first write the, okay, before I go to the box method, let me tell you the number here. So you can find 9 are there. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. Then there are 3 green marks, then 3 blue marks, and finally 1 circled green, wrinkled green. So is your result. Phenotypic ratio was the result. If you want to go for genotypic ratio, then you have to write each one separately. So you can find this is only 1. This is 2. This is again 2. These are 4. So how much is done now? 1 is to 2 is to 2 is to 4. Then you can find 1 again. So is to 1. You can find these 2. Then again 1. These 2. And again finally 1. So if they ask, maybe it won't be asked because it's not there in the textbook. The question, if it is asked, give the genotypic ratio of the dihybrid cross. So you have to write 1 is to 2 is to 2 is to 4, 1 is to 2 is to 1 is to 2 is to 1. If you are finding it difficult, you don't have to worry. This method will help you a lot. Of course, for CET this is important. So it is easy to memorize. 1 is to 2 is to 2 is to 4. You have to look at the box and you'll get the idea better. Okay. 1 is to 2 is to 2 is to 4. 1 is to 2 is to 1 is to 2 is to 1. I know it is going above your head. Let me tell you now about the how to write the box. Please remember, first write the capital. Okay. So for this, it may be the Y that is capital. It may be the Y which is recessive. Then there is second type, the R and small r and small r. Now again it is capital and small y. Now whatever I've done, these are the gametes of your F1 progeny. I'm talking about this. So how do you write this? It is a hybrid, right? So all four types are there. So it is first you write capital R, which has the possibility of going with the capital Y or small y. Okay, similarly there is your small r. Okay, so small r has the possibility of going with capital Y or small y. So this is how you have to encircle all the gametes. Once you have got that, first always remember to write capital R, then in front of it write capital Y and small y. Then you write small r, in front of that capital Y, small y. So I hope you are getting the idea. Similarly you are following in this direction. Okay, then only you will get this kind of checker box. So if you simply randomly write, here, you will not get this method. Okay, this is an easy method. 1 is to 2. So these 2 is to 3. Sorry, I'll repeat. 1, 2, 2, 4, 1, 2, 1, 2 and 1. I hope it is clear. So remember that triangle I had showed. This is your together 9. This is 3. So round and green is 3. Round and yellow. Okay, round and yellow is 9. Then wrinkled yellow is 3. And finally the wrinkled green, which is 1. So that is what is given in your textbook already here. You can follow that. Okay, this is the explanation. Okay, you can go through in your own time and you can include that. So finally what does it indicate? Who is dominant? Round and yellow. Because in your F1, though it was hybrid and you have got 16 varieties. Okay, you have got 16 varieties. Out of 16 you are getting 9, 3, 3 and 1. Okay, so please remember that. Do not worry about what is this 1/4. Do not worry. Okay, that this is very detailed explanation here. You have to know this diagram, not diagram, the schematic representation. These, all these seeds you need not draw. Now we are going for the next. The third law of Mendel, that is law of independent assortment. When two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. So you have already understood. There is one pair, another pair. So when they have to segregate in the gamete, one is R and Y, another gamete you will find R and Y. I hope it is understood. So if it is your hybrid, you know the 4 types. There could be chances of capital, small. Okay, so that is how we say R with capital Y, R with small y. This is how the segregation. It is independent, doesn't depend on the other pair of character. Okay, similarly, for small r, capital Y can come or small y can come. So this is how we say for the gamete formation, how it segregates during the gamete formation, how the hybrid segregates. Okay. So the chromosomal theory. Now we are coming to a important concept. Now till now whatever we studied was totally mathematical. So you have understood about ratios, you have drawn Punnett square, you are guessing that this is the cross, this is the feature, phenotype, genotype we are saying. Guessing, we are not giving the genotype. Genotype is the accurate. Phenotype is the guessing work, because we are unable to identify looking at the plant whether it is the hybrid or the heterozygous or homozygous or pure. So this is what we are saying. We have taken a mathematical logic here. Now we are going to the actual biological proof. When I say biological proof, Mendel, as I said, he was not a biologist. He had the interest over botany. Okay, since he had a garden, he was really, thrilled to study on the pea plants. Okay, so in a small area itself he could grow large number of plants, and he got good results within the time period, a shorter time period. So this is what was beneficial to him, and he gave his statistical record. But this is the person, Gregor Johann Mendel. So whatever his work was there, the biologist was not accepting it. So why it was not accepted? Because it is so difficult, and you know very well, the moment you are studying maths, sometimes it goes above your head or doesn't enter your head at all. Okay, so this is the same condition. The biologists were not able to take it. Okay, though his idea was clear, there was some communication problem. Okay, there was a communication problem, and he did not have complete proof. He had the proof of only a data which is written in maths. Statistical data he has. He doesn't have the actual proof of saying this is the character. He cannot show the character because it is present inside the organism. He can just write this is expressed, but he cannot prove it. So he is supposed to prove where that character is present. So at that time, about chromosome, he did not know at all. He did not know something there was present inside the cell that is carrying the characters. But he assumed it. He assumed there is something. There is something called factors. He termed it as factors. Okay, that was passing on to the generations, as it descends down. So as we have to look here, the dates, the year, if you see, he worked in the year 1865, but till 1900 his work was not recognized. So there are several reasons. Let us see why it was not recognized. First, communication was not easy. Now it is easy, definitely. Okay, then his work was not widely publicized. He could not publicize because there were very lot of restrictions, there were a lot of criticism. And the moment there is something that you all want, at that time, if there's anyone who want to prove an invention or discovery, it was very difficult to bring that particular possibility, or you can say making the audience know the reality of what the study was. It's very difficult to prove that reality. Okay, so it is immediately mocked by the audience by criticizing the person who has worked or researched. So this is a problem. Communication was not that easy, and that's how we did not publicize. So it did not go further for that reason. Second reason, he said about the factors being stable and discrete. Okay, but he couldn't explain what was the variation that was occurring. So variations were observed. The variation part he could not explain using statistics. He could not. Okay, so it is not 100% answer. That's why this, the all these laws are only basic but not acceptable, because it is only acceptable when there is no variation. Since the variation occurs, that's why we say we cannot accept it. Okay, so his concept that the factors are stable is not acceptable. Okay, he has not explained where the alleles are blending. Okay, and when I say blending, we are talking about the pink intermediate. Okay, and here he is the main reason why he did not get is, see, the stable and discrete units that control the expression of traits and of the pair of alleles which did not blend with each other. This concept was not accepted. The reason when we are saying it is not accepted, he's just given capital T and small t as not blend, but whereas capital R and small r has shown the blending. So what could be the reason? Give us a proof. He could not explain that. Okay, he could not explain that. So you have understood, I hope, because he's saying concept of gene is stable, but still in nature you can find variation. Then the third reason is mathematics explaining biological phenomenon was totally new and unacceptable. Okay, now finally you should know, of course it is all biological proof, but the audience were demanding for physical proof, what they are made up of. Okay, so biologists wanted a physical proof. Later on, these three people, Hugo de Vries, Karl Correns, and Erich von Schamack, I've written the names here, de Vries, Correns and von Schamack. These three scientists took Mendel's work of inheritance, and these three were not together. Okay, in that year 1900, they came to know that Mendel did some work, and he could not achieve it because of the certain drawbacks. So all those 4 points, these 4 points are your drawbacks. All these are your drawbacks. Now these three independently worked on Mendel's work and rediscovered the inheritance of characters. So what is that rediscovery? They could explain a little further what he was trying to tell. Okay, so they could do it. It was possible to do because by that time advancement in microscopy was present. Now we have microscopes. In his time microscope and all was not there. So that was one, there is one weak point where Mendel lost. But Mendel is still honored for his great work. He initiated this idea. Okay, so this microscopy helped them to observe the cell division. So it is very microscopic, and that is the proof we are getting now. So as the cell division was occurring, it led to the discovery of the nucleus structure. So how the cell was dividing, how the doubling of chromosome occurs, all that was observed. And very importantly, the term chromosome was given, and the chromosome, it was termed based on the staining method. So it is called as colored body as visualized by staining. So chromo means color, zomes means body, so colored bodies. So I hope the word is clear. 1 mark question, what are chromosomes? Now this chromosomal theory was initiated by Mendel and then modified by the three scientists, De Vries, Correns And finally, it was Walter Sutton and Theodore Boveri. These two people had given, using the Mendel's theory, they have given an acceptable theory with the proof, and with the present technology they could give the proof, and that's how we follow this theory compared to the Mendel's theory. Mendel's theory is always an important step for basics. Okay, now this person I've kept here, he'll come bit later. Okay, so chromosomal theory proposed by Walter Sutton and Theodore Boveri. Remember, chromosomal theory of inheritance is also called as Sutton and Boveri theory of inheritance. Then you should know in which year. So in 1900, three scientists worked on Mendel's work separately, individually, and then they gave their ideas. Okay, so by 1902, these two scientists together, Sutton and Boveri together worked, and they studied the chromosome movement during meiosis. Very important. They studied the chromosome movement during meiosis. So what is that chromosomal theory of inheritance? 5-mark question. Sutton united the knowledge of chromosomal segregation with the Mendelian principles. So whatever 3 laws you remember, those are your Mendelian principles. And uniting the knowledge of chromosomal segregation, that is how the movement of chromosome occurs. So together with the Mendelian principle, he brought forward chromosomal theory of inheritance, a widely accepted, universally acknowledged. There are the 3 points you should remember for the theory. The behavior of the chromosome was parallel to the behavior of genes. Why? If the gene has to pass on to the gamete, then it is present on the chromosome, right? So if the chromosome is also going one way, that means even the gene is going to the same direction. It carries the gene along with it. That is why we say behavior is parallel. Then the chromosome as well as genes occur in pairs. So as I said, in diploid organism we have your homologous, that is paternal, maternal, and each will have the gene on it. So imagine A. So here also the gene is paired. Even chromosome we are saying a pair. So they occur in pairs, clear? 2 alleles of a gene pair are located on the homologous sites. Now when I say homologous sites on homologous chromosome, again you should know what is homologous chromosome. They are duplicated. Here also homologous, duplicated. So I am saying about, so imagine this area, okay, this area is the gene A. So I hope you understood. The position I've marked on the chromosome, this is your homologous paternal and maternal chromosome. Paternal, maternal. So once you are able to get those two, so this means 2 alleles of a gene pair. So A. Even on the other one, A. So once you have understood that part, we are saying they are present on the homologous sites on homologous chromosome. So during anaphase, the 2 chromosomes, the homologous chromosome, will first align at metaphase plate independently and then segregate. Okay, so when I say independently, you have to follow the textbook diagram. You can see how it is duplicated here. Then there is your metaphase stage, meiosis 1 anaphase, where the separation takes place. Then meiosis 2 anaphase, where chromatids of the chromosome separates. This is homologous separation. This is chromatid separation. observe, here there is only one type they have shown. You can see here, which I am explaining, a little different in I would say almost like a graphical representation. So you can observe here, this is how Mendel explained A is separating, but he did not know what was that carrying them. So it was later on understood it was chromosome that was carrying the factors, which is called as genes. So we are saying independent assortment. So there is separation here. Look at the color. It can be paired with the dark one, or it can be paired with the light one here. So that is what we are saying, they are independently assorted. Okay, so you get the better idea here. So I hope it is clear. Now there is experimental verification. Now when I say experimental verification on chromosomal theory, we need a proof and someone to verify it is true. So it was Thomas Hunt Morgan, or T.H. Morgan, along with his colleagues, he discovered the basis for variation. Very important. Now we are talking about the variation. When does variation occur? During sexual reproduction. So what specimen did he choose? You know this person? Yes. It is a fruit fly. Scientific name is Drosophila melanogaster, underlined separately when you are writing. So this was very suitable for studies. Okay. Now this particular fruit fly is called as the Cinderella of genetics. Okay, Cinderella of genetics, and it was so useful and very easy to handle. So you'll understand why he selected especially among the all organisms, he selected fruit fly for his study. Okay, why? Because it had all the easy characters for him to manage during his study. Let us understand what are those characters. One, they are grown on simple synthetic medium. You can grow on any sugary material, anything that is decaying. So you can prepare an easy medium for it to grow. So you know how they lay the eggs and the maggots come. That is how it works. Then there is your life cycle that completes in about 2 weeks. So it's very short period, you get a very soon result. Okay. So compared to Mendel only he worked, but his work was a little faster. And single mating was producing large number of progeny flies. So one mating is enough to produce large amount of offsprings. Even the sexes can be identified, that is the male and the female can be easily identified. So they are distinguishable. There are many types of hereditary variations. This helped him to bring the study of variation concept in his research. So, and all these hereditary variation he could study under the low power. You remember the dissection microscope? In that only you can identify, looking at the eye color, the wing shape, body color, okay, body size, all that you can easily identify there. So those are your hereditary variations. So what did Morgan do? He conducted several dihybrid crosses, not monohybrid, dihybrid crosses. Okay, and especially all those were sex linked. His studies were sex linked. When I say sex linked, we are talking about chromosome X, chromosome Y. Now when Mendel did, I hope you all know, he did it on the pea plants. So he compared his study and did it on Drosophila. So what was that he had selected? He selected yellow body, white eye females. Okay, body is yellow but eyes were white, and those were females. He selected the females with that character. So 2 characters are there. Then crossed with brown body and red eyed males. So which is dominant here? Yellow and white. So they were crossed. Okay, after crossing, he could see that his F2 generation, okay, F2 generation was not as expected in the ratio 9:3:3:1. It was not that ratio. It was a deviation. So what was that deviation? It means the two genes did not segregate independently. They were depending. This concept of dependence of genes, that means the genes need to be each other with each other. So what was that reason? He understood whatever he was studying was on the chromosome X. So these two genes were situated on the same chromosome, whereas Mendel, he selected two different chromosomes, saying that R is here, Y is on the next chromosome. But in case of what Morgan has found, he found the genes are present, the characters were present on the same chromosome. So whatever the pair of characters he chose, one is the body color and the eye color, it was on the same same chromosome, on the X chromosome. So what does it show finally? So in F2 generation, he is saying parental gene combination was much higher than the non-parental combination. So you will understand what is that parental and non-parental. So he described, let me show you this picture here. So you can see whatever here it is, the white, yellow and white. You can see here. So remember, we show female, okay, as straight, whereas males will show one this method. So whatever like a hook like structure you find, it is Y chromosome, and that's why we call the sex chromosome in case of males as hemizygous. Okay, so let's go a little further. What we are trying to understand here is the what has happened to the two pairs of characters. So you can find they are closely linked. So as they are coming forward, you can find parental combinations are more than the non-parental. What is the non-parental type? We are saying the cross. So here you may not understand because we are not going deeply in that study. Okay, we are just giving you the idea. Parental gene is much higher than non-parental type. So this is actually your parental type. This is parental type. Yellow body, white eye, brown body, red eye. Those are your parents in Drosophila. But what happened? A deviation occurred which led to the difference, that is parental gene combination is more and the non-parental type was less. So that was a deviation from the dihybrid cross from the Mendelian's third law, Mendelian's principle. So he, based on his work, he gave two terms, linkage and recombination. So when I say linkage, we are saying the physical association of the genes on the chromosome. It can be two or more than two genes that is linked. They need to be or depending upon each other during segregation. Then there is recombination, exchange of gene. To understand what is that exchange of gene, you have your chromosome. Okay. So imagine this is the portion. Now what happens is this portion gets interchanged. What happens? It gets interchanged. Let me show how is that interchanging. So I hope you got it. This portion, which was pure, has got interchange. You can see the color changes. The segment has got exchanged. Segment of DNA, segment of the chromosome is got exchanged. This is called a new combination, recombination. I hope you got some idea. Okay, so it is trying to describe the generation of non-parental gene combination. I hope you understood now. Because of the exchange of the segments of DNA segments, there is non-parental gene combination. Okay, so this is what they are trying to explain. So this is your two types. You can see how far they are and how close. In the second one, you can find the white and the miniature wings. So it's explained here. You can find, this is from the textbook I have taken. So what did his team do? They found out when the genes are grouped on the same chromosome, that is linked on the same chromosome, there are two types of linkages, tight linkage as well as loose linkage. If it is tightly linked, there is low recombination. Okay, so imagine if this is the chromosome and they are closely located here. The chances of them to separate is difficult. Now in case if there is a chromosome here and there is very far linked genes, then the chances of them to be exchanged is high. So that is what we say. If it is far, it is loosely linked, so higher recombination. If they are closely linked or tightly linked, they have low recombination. It is difficult to separate them. Here it is easy to separate them for the exchange. Okay, so you have got some idea. So that's how they have given the percentage. So these 2 points are important for the experimental verification of Morgan's study on chromosomal theory of inheritance. So this is all Morgan's study showing linkage, recombination as well as genetic mapping. What is genetic mapping? One of his student, Alfred Sturtevant, he used that frequency of recombination. What is that frequency? You can find here 1.3% recombination occurs. 37.2% recombination occurs. This is the frequency. If it is tight, only 1.3%. If it is loose, okay, we are saying about the more percent of recombination. This is what we are saying. So looking at even at the picture, you can say if far, higher chances of recombination. If they are close, less chances of recombination. I hope you got the idea. So based on that, he could measure the distance between the genes and immediately map the position of the chromosome. Position of the gene on the chromosome. Position of the gene on the chromosome. So I hope you understood. The distance is understood by measuring, understanding the frequency and then measuring, and then once the position is fixed, it is mapped. This concept is called as genetic mapping, brought forward by Alfred Sturtevant. So I hope it is clear. So this is what you can just understand a little more they have given. This is what I wanted to say about hemizygous condition and the homozygous female. So this is your sex chromosomes we are dealing with. Highest variable characters are seen in sex chromosomes of Drosophila. So you can just go through this. This is again, if you are finding it difficult, you can skip. So we are almost done with this particular portion of chromosomal theory. Okay. So next we will be studying about chromosomal disorders. If there are any doubts you have, let me know. So please go through the meiosis topic and then understand this topic again. Okay, it's very difficult to understand chromosomal inheritance. Yes, thank you.